What is nanotechnology? Nanotechnology is technology on the nanoscale, 1 to 1,000 nanometers, depending on what definition you look at. And what does it mean to be on the nanoscale? So, for instance, a human cell is 30 micrometers, that's 30,000 nanometers, so a lot larger than what we would consider nanosized. However, the organelles inside the cell, they're examples of nanosized biological structures. Then, for instance, bacteria, 500 nanometers, and viruses, 50 nanometers, are examples of biological nanoparticles. So what is really the advantage of utilizing technology on the nanoscale? There's two major benefits. The first is that we get higher functionality in smaller spaces. And the second is that we get these unique electromagnetic properties that are not apparent when a material is in the form of a small molecule or as bulk matter. And nanotechnology has revolutionized several fields, including the computer industry, where these computer chips have become smaller and more efficient. Another example is solar power technology, where the individual units in solar panels have become smaller and more efficient. And then when we talk about medicine, most conventional drugs are small molecules, so 0.1 nanometers. And then several decades ago, we started using antibody therapeutics. And now we even have several nanoparticles that are clinically approved on the market. But on the contrary to these two first examples, when we talk about medicine and nanotechnology, we're actually going larger. So what are the specific advantages of going larger, or in other words, what are the benefits of nanomedicine? And the first is multifunctionality. So for instance, instead of having one drug, we can have multiple drugs in the same nanoparticle. The second is these unique electromagnetic properties that I already mentioned, but we can also utilize these properties for therapeutic purposes. And the third advantage is that they are differently processed by the body. And this has a major impact on the final location of the particles inside the body, which then affects therapeutic efficacy and safety. And I'll talk briefly about all of these three, and I'll start by discussing multifunctionality. And here you see a schematic of a multifunctional nanoparticle. So there's several additional supportive functions we can add with nanomedicine. And the first example is solubilization. So there's several chemotherapy drugs that are not water soluble. So in order to administer them to patients, you need to use toxic solvents. So another approach to avoid this is to use encapsulation of these drugs inside a nanoparticle. And one clinically approved example is Abraxane, which is albumin bound to paclitaxel that forms a nanoparticle. And it's approved for several cancers, for instance, pancreatic cancer. The second example of multifunctionality is that nanoparticles can protect drugs from degradation. And this degradation can be mechanic or enzymatic. And this is especially important where we're dealing with therapeutic agents that are very sensitive to breaking down, such as small interfering RNA. And a clinical stage example is A2O27. This is a lipid-based nanoparticle with small interfering RNA inside. So this is a gene silencing agent, and it has completed phase 1 and 2 clinical trials. The third example is immune evasion. So we can modify the surface of nanoparticles so that they are hidden from the immune system. And this is mostly done with various polymers. The one that is used a lot in the clinic is polyethylene glycol, so pegylation. And a clinically approved example is Doxil. So this is a lipid-based nanoparticle with doxorubicin inside. And on the surface, it is pegylated with this polymer. And this polymer basically prevents opsonins in the blood circulation from binding to the surface of the nanoparticle, and these opsonins in turn would trigger macrophage uptake of the nanoparticles. Then targeting is the fourth example. So we can conjugate targeting ligands to the surface of nanoparticles, and these targeting ligands can recognize specific receptors on diseased cells. So a clinical stage example is MM310. So this is an FRNA2 targeted lipid-based nanoparticle with docetaxel that is currently in phase 1 clinical trials. Then we can also include imaging agents together with therapeutic agents in the same nanoparticle. Currently, there's no clinical stage examples, but a lot of preclinical work has been done looking at theranostic particles that are both therapeutic and can be visualized inside the body. Then we have nanoparticles that contain permeation enhancers, which actually help the nanoparticles with the drugs get throughout the tissue, so permeate the entire tissue. Again, a lot of preclinical examples, they have not reached the clinic yet. Then we have triggered release strategies. So these nanoparticles are activated in response to external signals in diseased tissue. A clinical stage example is CX-2009, which is a CD166 antibody that is conjugated to a cytotoxic agent through a peptide linker. And this peptide linker can only be cleaved in the tumor microenvironment, and once it's cleaved, it can have a cytotoxic effect to the cells. And then what I already mentioned, the last example, is combination therapy, so having several drugs in the same nanoparticle, and Bixias is a clinically approved example for it has cytarabine and daunorubicin inside a lipid nanoparticle. So as you can see, there's several ways we can utilize multifunctionality when it comes to nanomedicine, and most of these are already in the clinical stage or even clinically approved. And within my own research, I've also worked with multifunctional nanoparticles, for instance, silicon particles, and we've looked at encapsulating multiple drugs, for instance, chemotherapy and gene silencing agents in the same nanoparticle. We've also looked at sequential drug release strategies, where drugs are released at specific times in specific locations inside the body. We've looked at stimuli-responsive drug release that you already heard about on the previous slide. I've also worked with liposomes, which are these lipid-based vesicles that we can load drugs inside. And we've looked at encapsulating multiple drugs, chemotherapy, together with natural compounds. We've looked at shielding from the immune system using various polymers, and also targeting ligands. So in one example, we conjugated thyroid-stimulating hormone to the surface of these nanoparticles and used this to treat thyroid cancer in a mouse model. So we get higher accumulation in the thyroid due to this targeting ligand. So the second advantage is these unique electromagnetic properties that appear. And these can be optical, magnetic, electronic, or thermal. And the take-home message is that they're not apparent on the molecular or macro scale, but only on the nanoscale. And the reason for this is that nanoparticles have a large surface area to volume ratio, and this causes unique properties to arise. And in addition to this, they're also similar size as the wavelength of light. And again, this causes unique characteristics to appear. So within my own research, I worked with gold nanoparticles injected into the tumor. We shine an infrared light, which makes these gold nanoparticles heat up and basically burn the cancer cells. But there's also a clinically approved example of utilizing the electromagnetic properties of nanoparticles for therapeutic purposes, and this is Nanotherm, is the product, and the company is Magforce. It's clinically approved in Europe for the treatment of glioblastoma. So basically what they do is they inject iron oxide nanoparticles locally into the brain of glioblastoma patients. Then they put these patients in a magnetic field. This makes the iron oxide particles heat up, and basically the thermal elevations, temperature elevations, causes the tumors, the cancer cells to die. And then the third example that says number 1, but is supposed to say number 3, is the fact that nanoparticles are differently processed than small molecules and conventional drugs. And I'll spend a lot more time talking about this third benefit because I think it's more complex to understand than the other two. So when we talk about site-specific delivery of systemically administered drugs, so that's either taken orally or injected intravenously, we know that for conventional drugs, only 0.001 to 0.01% of the drug will reach the intended location. So these values are very scary, especially if it's the first time you see them. So with nanoparticles, we get a substantial improvement. Around 1% of the nanoparticles that we inject intravenously will actually end up in diseased tissue. And because of this improvement in biodistribution, we typically see that nanoparticles have increased therapeutic efficacy and reduced side effects. But one of the questions we asked is 1% enough? And how can we further improve site-specific delivery? And within the field of drug delivery, we believe that treatment of disease is fundamentally a problem of drug distribution. And the ultimate goal is to exclusively direct and confine therapeutic agents to diseased tissues. And we know that all drugs have this therapeutic window, which is a range of drug concentrations. And if you go below it, you don't see any therapeutic activity. And if you go above it, you get side effects. But if we could find a way to specifically direct drugs to diseased tissue, then we could expand this therapeutic window and eliminate the upper threshold. And here on the next slide, you see a simplified analogy of drug delivery, where the car represents the drug delivery system and the road, the biological environment. And in the middle, you see this fire pit that symbolizes the biological obstacles in the body that prevent the drug from going where it's supposed to. So in order to improve drug delivery, we can either upgrade the drug delivery system, and we have several examples doing this in the lab, or we can change the biological environment, which is a less common approach, but something that I'm very passionate about. But I'll start by discussing the design of these sophisticated drug delivery systems. And the thing we really try to do is take advantage of differences between healthy and diseased tissue. So for instance, as an example, if we take a look at cancer blood vessels, they differ in two distinct ways from normal vasculature. The first way is that the blood flow is slower, and this is because as a tumor grows, a vasculature network needs to form very quickly to support this tumor growth, and because this process is quick, it's disorganized, and this disorganization causes the blood flow to be rather slow. The second difference are these fenestrations or holes. And again, this is because the vasculature forms quickly, it's immature. So it doesn't really have time to form this continuous barrier. So we asked, can we take advantage of this? And the answer was yes. So to take advantage of the slower blood flow, we designed disc-shaped microparticles. So these were around 1 micrometer in diameter, so 1,000 nanometers. So an example of a large nanoparticle. And then to take advantage of the fenestrations, we designed smaller nanoparticles. And on the next few slides, I'll illustrate how each of these particles interact with the vasculature. So we know that these microparticles, due to their size and shape, tend to flow very close to the vessel wall instead of in the middle of the blood vessel, and this is due to fluid dynamics. So we've done mathematical modeling and experimental studies to find the optimal shape and size for the nanoparticles to flow close to the endothelial cells. And once they are in close proximity to the endothelial cells, they will adhere to the surface because they have this large parallel surface area. So if we take a look at what happens in cancer vasculature, the microparticles will bind to the vessel wall, and then they will actually stay bound to the vessel wall. However, in normal vasculature, the microparticles will still bind because they're designed to do this, but because the blood flow is faster, it will cause the particles to dislodge. So here the dislodging force is higher than the attachment force. So this means that we get higher accumulation of these disc-shaped microparticles in tumors compared to healthy vasculature due to fluid dynamics. And if we compare this to conventional drugs, we know that differences in blood flow doesn't really impact small molecule accumulation. So we don't see a site-specific tumor delivery. So this is a way we can use nanoparticles or microparticles to achieve enhanced tumor accumulation, which is not possible with something that is a small molecule. And this is just a schematic showing what I just illustrated, that these discoidal microparticles flow close to the vessel wall, and then in tumor vasculature in the lower panel, you can see that they actually stay adhered to the surface. And the interesting thing is that 99% of clinical and preclinical work on nanoparticles is actually spherical nanoparticles, but this shape is not optimal at all, as we can see from tumor studies. So then when we take a look at the nanoparticles, we know that these are smaller, so they can enter through these fenestrations or holes in the vasculature wall. And this is what happens in cancer tissue. However, in normal vasculature, we don't have these vasculature holes, so we don't see any nanoparticles entering into the tissue. So again, this is a way to achieve enhanced accumulation of nanoparticles in tumors versus healthy tissue. And again, if we compare this to conventional drugs, small molecules, we know that blood vessel fenestrations don't really impact small molecule accumulation because small molecules are so small that they can diffuse everywhere throughout the body. So now we'd utilize two different approaches to achieve higher accumulation of drug delivery systems in tumor vasculature. So we thought, why don't we combine these two approaches? And so we decided to take disc-shaped microparticles using porous silicon microparticles and load them with nanoparticles inside. And this platform we named the multi-stage vector because it has multiple stages. So after we inject this MSV, multi-stage vector, into the blood, the microparticle will bind to the blood vessel wall. Here you see a scanning electron microscopy image. You see the multi-stage vector bound to the endothelium, and then you see these characteristic fenestrations that are present in tumor vasculature. And then the microparticle will slowly degrade because porous silicon is a biodegradable material, and as it degrades, it will release these nanoparticles that can then enter through the fenestrations in the vascular wall. And we've used this platform, this multi-stage vector, to treat several conditions, several cancers, and one example is melanoma lung metastasis. So in this case, our goal was to achieve high accumulation in the lungs. And so when we compared liposome accumulation, so conventional nanoparticle, to liposomes loaded inside the multi-stage vector, we saw that lung accumulation was substantially increased. And this is the quantitative analysis. In blue and black, you see the multi-stage vector. We saw the same trend when we compared polymeric nanoparticles, a spherical nanoparticle, to the multi-stage vector. Again, we saw enhanced lung accumulation. And what you don't see in these images are the free drug, but I can tell you that we wouldn't see any accumulation here if we're looking at a conventional therapeutic agent. So then we wanted to see whether this improvement in biodistribution could translate into better therapeutic efficacy. So we did a mouse experiment. There's a lot of control groups here, but this last group is using the multi-stage vector, and we can see that tumor burden was reduced. The survival of the mice were also increased, and the lowest number of metastatic nodules in the lungs in the multi-stage vector group. So this is really an example of how we can create these sophisticated drug delivery systems to improve site-specific delivery and therapeutic efficacy. And so in regards to clinical translation, this work was done at the Houston Methodist Research Institute, and this is a picture of that building. So in the 12th floor, we have a current good manufacturing practices facility, which is required by the FDA. And in order to get FDA approval, you need to show that you can produce a high enough quantity of your therapeutic agent. And that's what we've been doing. And we got a Department of Defense grant of 16 million. And the goal of this grant, it's called Breast Cancer Breakthrough Award Level 4, is to take this platform into phase 1 clinical trials. So that will be happening in the coming months. The lead on this trial is Dr. Mauro Ferrari, who's also my previous PhD mentor and the CEO and president of the Houston Methodist Research Institute. And for this particular trial that we got the funding for, it's for breast cancer lung metastasis. And this is a very unique way to fund a phase 1 clinical trial. Usually we see the research done at academic institutions, non-profit institutions, go to big pharma for phase 1 clinical trials, but we're trying a different model here. And we also have recently got around 20 million philanthropy funding to further support phase 1 clinical trials at the Houston Methodist Research Institute. So I want to conclude this part of the work before I move on to the next part. So we talked about the 3 advantages of nanomedicine: multifunctionality, solubilization, protection of the drugs from degradation, shielding from the immune system, adding targeting ligands, imaging agents, permeation enhancers, trigger release, and finally combination therapy. We talked about unique electromagnetic properties that appear on the nanoscale. A clinical stage example is approved in Europe with iron oxide nanoparticles for glioblastoma, where you put the patients in a magnetic field. Then we talked about the fact that nanoparticles are differently processed by the body than conventional drugs. And we can exploit this slower blood flow in tumors by designing discoidal microparticles, and we can exploit the tumor vasculature fenestrations by designing smaller nanoparticles. And if we put these 2 strategies together, we can come up with the multi-stage vector concept that is now entering phase 1 clinical trials. So this is an example of a nanoparticle design approach to improve site-specific delivery. But as I mentioned, we can also modify the microenvironment to improve drug delivery, and this is what I'll be talking about next. So within my own research, I worked a lot with strategies to modify the microenvironment, for instance, deactivation of liver macrophages, anti-tumor immune responses, modulation of the innate immune system, complement proteins, and modification of the tumor microenvironment. But because time constraints, I'll talk about one of these projects, which is deactivation of liver macrophages. And as I already mentioned, nanoparticles have improved site-specific delivery compared to conventional drugs. Around 1% of nanoparticles that we inject into the blood will actually reach the tumor. What I didn't mention was that up to 99% will go to the liver and spleen, and this is due to resident macrophages in these organs that are designed to recognize and engulf foreign matter, such as bacteria and viruses, which are nanoparticle sized. And they form the mononuclear phagocyte system. So in a hypothetical example, if we could block the liver and spleen by, for instance, 1%, that's not a lot. In the best case scenario, we could increase tumor accumulation by 100%. So a small change in what goes to the mononuclear phagocyte system could have a major impact on therapeutic benefit. So we thought, what if we find a macrophage switch to temporarily block the activity of macrophages in the liver and spleen, thus preventing them from being able to uptake nanoparticles? And so we decided to go with a drug repositioning approach. So we asked whether there were drugs that are already clinically approved that could perhaps inactivate macrophages. And so we did a cell-based screening of a bunch of different drugs, and we found that chloroquine, an anti-malarial drug, was the most effective at blocking nanoparticle uptake in macrophages. And we looked at several different nanoparticles. I'm showing you one example, which is liposomes, lipid-based nanoparticles. And here's 3 different macrophage cell lines, RAW2647, J7748, murine macrophage cell lines, and then Kupffer cell lines, which are the resident macrophages in the liver. And we see that already after 3 hours in control cells, these macrophages had taken up a lot of fluorescently labeled liposomes. When we pre-treated the cells with chloroquine, we found a statistically significant reduction in liposome uptake, and we made sure that these concentrations that blocked nanoparticle uptake were not toxic to the cells. But then, of course, we wanted to make sure that chloroquine was not blocking nanoparticle uptake in cancer cells, because, of course, this would be counterproductive. So we looked at 3 different cancer cell lines, MDA-MB231 breast cancer cells, MIA-PACA2 pancreatic cancer cells, and H358 lung cancer cells. And we saw that chloroquine didn't really affect liposome uptake, suggesting that perhaps this is a macrophage-specific response. And then we wanted to see what would be the effect of chloroquine in vivo in a mouse model. But for this, we wanted to determine a clinically relevant dose. So we did a dose translation from human to animal studies based on FDA guidelines that take into account body surface area. And the standard human dose for malaria treatment in the clinic is 2.5 grams of chloroquine, and this corresponds to a mouse dose of 514 milligrams per kilogram. But for practical reasons, we were doing intraperitoneal injections in the mice, and in humans, this drug is given orally. So we also wanted to take into account the bioavailability, which is how much of the drug is absorbed by the intestines. So for chloroquine, that's 89%. And so we recalculated this, and we ended up giving a dose to the mice that was lower than the standard human dose. So then we looked at the accumulation of liposomes in control mice at various time points in the plasma, liver, and spleen, and the goal was to find a time point where liver accumulation was higher than plasma accumulation. Because if we're studying liver uptake, we want to make sure that most of the liposomes are not in circulation. And we found that this was at 6 hours. So we chose to work with this time point, and we also used a positive control for our studies, which is always important in science. And this is a macrophage depletion agent that has previously been described in the literature. And when you give this agent to mice, 24 hours later, all the macrophages in the liver are eliminated. And this is not a clinically relevant approach because it's toxic, but it served as a positive control in our studies. So the mice that were pre-treated with the macrophage depletion agent had a higher plasma to liver accumulation ratio. So a lot less liposomes were in the liver, a lot more in the plasma. So this illustrates that the macrophages really play a role in nanoparticle biodistribution. Then when we pre-treated the mice with chloroquine, we saw a similar effect. We saw higher amounts of liposomes in the plasma and less in the liver and spleen. So next, we wanted to see how chloroquine affects tumor accumulation, and for this, we used an MDA-MB231 triple-negative orthotopic breast cancer model. And we did the biodistribution studies when the tumors were rather small, but they still showed a vasculature. And this is after 6 hours, so we can see that only 0.7% of these lipid nanoparticles actually reach the tumor, which illustrates that, again, the liver and spleen are major barriers for nanomedicine. And when we gave the mice the positive control, we saw that tumor accumulation increased, and the same trend with chloroquine. So now we've shown that chloroquine pre-treatment works for one type of nanoparticle, liposomes, for one type of condition, cancer. So next we asked whether this strategy could have broader applications. And for this, we decided to work with the discoidal silicon particles. And we've also found that if we further tailor the size and shape of these particles, we can make them go to certain healthy organs. So in this case, if we use microparticles that are around 2 micron in diameter, they will go to healthy lung vasculature. So we asked, can chloroquine pretreatment improve organotropic accumulation of these silicon particles in the lungs? And indeed, we found the same trend as with the liposomes. We had higher accumulation in the blood, less in the liver, and indeed we did find that lung accumulation was increased, showing that chloroquine works for 2 different nanoparticles in vivo and also for 2 different conditions. One is healthy tissue and one is cancer. Next, we just wanted to make sure that chloroquine was not killing the macrophages in the liver and spleen, because this would be toxic, so we did some staining. In green, you see the macrophages, in blue, the nuclei, and as you can see, with the macrophage depletion agent, all the macrophages are gone from the liver, and there's a slight reduction in the spleen, but the chloroquine groups looks very similar to the control group, indicating that this is not a macrophage killing strategy, but a macrophage deactivation strategy. And then, of course, we were interested in understanding the mechanism by which chloroquine was blocking macrophage uptake of nanoparticles. And so we did some mass spectroscopy analysis, and we found that distinct protein populations were upregulated in control cells and chloroquine-treated cells. And specifically, we found that all these proteins involved in endocytosis, which is the process that nanoparticles are internalized by cells, were only present in the control cells. And in particular, we were interested in phosphatidylinositol binding clathrin assembly protein, PCALM, because this protein is very important for clathrin-mediated nanoparticle uptake, which is one of the main mechanisms that nanoparticles are internalized. And the 3 most abundant proteins in clathrin-coated vesicles are clathrin, PCALM, and the AP2 complex. And previous studies have shown that if we suppress PCALM, we also inhibit clathrin-mediated endocytosis. So we wanted to confirm these results with the Western blot, and indeed we found that chloroquine does significantly suppress PCALM expression, but doesn't really affect the other proteins in the clathrin-coated vesicles. So perhaps this is one of the mechanisms by which chloroquine blocks nanoparticle uptake. And in regards to clinical translation, chloroquine plus any clinically approved nanoparticle could be given to patients today. This is something called off-label use. And this is also why we decided to go with a drug repositioning strategy, so that we could quickly get it into the clinic. We know that usually it takes 15 years and $1 billion to get something into the clinic. So we could combine chloroquine with any number of nanoparticles that are clinically approved. But we are looking at doing a potential clinical trial here at the Mayo Clinic, just to make sure that these mice studies can also be translated into humans. So we have some initial discussions with companies that produce clinically approved nanotherapeutics. So to conclude this section, the mononuclear phagocyte system is a major barrier for localized drug delivery. We can use pharmacological inhibitors of resident macrophages in the liver and spleen, for instance, chloroquine, through a drug repositioning strategy. And this reduces nanoparticle accumulation in the liver and spleen and increases tumoritropic accumulation of liposomes and organotropic accumulation of silicon particles in the lungs. And a potential mechanism is suppression of PCALM. So I want to acknowledge the people that made this research possible. So my previous mentors, Dr. Mauro Ferrari, Dr. Haifa Shen, and Dr. Elvin Blanco, and then our funding sources from Houston Methodist Hospital, NIH, Department of Defense, the Cancer Prevention Research Institute of Texas, and my personal funding from Victoria Stiftelsen and Nylands Nation in Finland.